Up the Down Staircase. Brainstem, Part 1. The brainstem consists of the medulla oblongata, pons, and midbrain. Inferiorly, it is continuous with the spinal cord, superiorly with the diencephalon. Posteriorly, the brainstem is connected to the cerebellum by the superior, middle, and inferior cerebellar peduncles. Several cranial nerves, from the 3rd up to the 12th, emerge from the surface of the brainstem. The medulla oblongata is divided into a lower, closed part, which surrounds the continuation of the central canal of the spinal cord, and an upper open part, which is related to the lower part of the 4th ventricle. The arrangement of gray and white matter in the medulla oblongata is different at various levels. In the lower part, the transverse section through the medulla oblongata is similar to that of the spinal cord. The central canal is surrounded by central gray matter. The descending and ascending tracts are present bilaterally. However, there are some differences. Firstly, the presence of extensions of the central gray matter which form the gracillae and cuneate nuclei. Gracillae and cuneate fasciculi terminate in these nuclei. Axons of second-order sensory neurons arising from the multipolar cells of these nuclei are termed internal arcuate fibers. They cross the midline and ascend on the opposite side as medial lemniscus. Secondly, the network of fibers and scattered neurons and gliocytes termed reticular formation. It will be described later. And thirdly, many fibers of the pyramidal tract crossing the midline, forming the decussation of pyramids. After crossing the midline, these fibers run lateral to the central gray matter and form the lateral corticospinal tract. The fibers which do not cross descend into the spinal cord and form the ventral corticospinal tract. In the transverse section of the upper part of the medulla oblongata, the ascending and descending tracts, spinal nuclei of trigeminal nerve, and reticular formation are present. Large compact bundles of nerve fibers form the inferior cerebellar peduncles, which house the dorsal spina cerebellar and olivocerebellar tracts, among others. The ventral spinocerebellar tract lies more anteriorly near the surface of the medulla. On the anteromedial aspect of pyramidal tracts, arcuate nuclei, the extension of the pontine nuclei, lie. They receive fibers from the cortex and send the axons via the anterior external arcuate fibers to the cerebellum of the opposite side via the inferior cerebellar peduncle. The dorsal part of the medulla oblongata at this level forms the floor of the 4th ventricle. In relation to the latter, nucleus of the hypoglossal nerve, dorsal nucleus of vagus nerve, nuclei of vestibulocochlear nerve, nucleus ambiguous, nucleus of solitary tract are localized. Another prominent structure at this level is the inferior olivary complex consisting of the principal olivary nucleus, medial and dorsal accessory olivary nuclei. The principal olivary nucleus has a laminar structure and in the transverse section has crumbled bag-like appearance. All olivary nuclei consist mainly of projection neurons that have a prominent arbor of spine-bearing dendrites. Dendritic spines of neurons are club-shaped with unusually long necks. Because of long necks, the spine heads can cluster together and form the core of what is the most characteristic feature of the olivary neuropil, the glomerulus. It contains several dendritic spines derived from different neurons and several nerve terminals covered with a glial sheath. The dendritic spines are usually electronically coupled by gap junctions, is a remarkable feature of the inferior olivary neurons which ensures the synchronous firing. Inferior olivary complex neurons receive inhibitory and excitatory inputs from the deep cerebellar nuclei and from mesodiencephalic junction. The latter includes various nuclei of brainstem, among which nucleus of Darkshevich, red nucleus, interstitial nucleus of Cajal, and superior vestibular nucleus project to the olivary complex. Axons of the olivary complex neurons form climbing fibers of the olivocerebellar tract which cross the midline, ascend into the cerebellum, and synapse on Purkinje cells. At the cross-section of pons, tegmental or dorsal and basilar or ventral parts are distinguished. In the basilar part, at all levels, pontine nuclei are localized. They receive fibers from the corticonuclear tract. Nerve fibers that arise from the pontine nuclei cross to the opposite side as transverse pontine fibers and run to the cerebellum via the middle cerebellar peduncle. Besides transverse nerve bundles, there is pyramidal tract longitudinal, which consists of corticospinal and corticonuclear tracts, with the latter ending in the motor nuclei of cranial nerves. The transverse section of the lower part of the pons passes through the nuclei of trigeminal, abducens, facial nerves, and nuclei of vestibular cochlear nerves. The white matter at this level consists of medial longitudinal fasciculus, medial and spinal lemnisci, and trapezoid body, which is a trapezium-shaped bundle of transversely running and decussating nerve fibers arising from the cochlear nuclei. The spinal lemniscus is mainly formed by nerve fibers of the lateral spinothalamic tract. There are four vestibular nuclei: inferior, medial, lateral, and superior. The medial and inferior ones are located in the upper part of the medulla oblongata, the lateral and superior ones in the pons. The medial vestibular nucleus contains many small to medium-sized, densely packed multipolar neurons and a sparse population of large neurons, giving rise to the bilateral medial vestibulospinal tract. It descends to the mid-thoracic segments of the spinal cord and mediates the head position by controlling the muscles of the neck and shoulders. The lateral and superior vestibular nuclei contain many small, medium, and large, darkly staining multipolar neurons. The neurons of the lateral nucleus give rise to the lateral vestibulospinal tract, modulating postural adjustments to movements. The superior and medial vestibular nuclei are connected with the extraocular motor nuclei through the medial longitudinal fasciculus. Afferent nerve fibers from primarily the superior vestibular nucleus ascend to the thalamus. Within the reticular formation beside the medial longitudinal fasciculus, or may lie within the lateral lemniscus. The inferior and medial vestibular nuclei are connected with the cerebellum through the inferior cerebellar peduncles. The inferior vestibular nucleus possesses many medium to large multipolar cells and is characterized by the presence of many longitudinally running nerve fibers of the lateral vestibulospinal tract, which give the nucleus a speckled appearance. The histological structure of cochlear nuclei has some peculiarities. They consist of identifiable neuron types with predictable synaptic relations. The ventral cochlear nucleus possesses bushy, stellate, and octopus neurons on which nerve fibers of the cochlear nerve form synapses. Bushy neurons, glutamatergic, have a few short, branched dendrites, making them look like a bush. T-stellate neurons are glutamatergic, and D-stellate neurons are glycinergic inhibitory neurons that have long, sparsely branched dendrites and long axons. Octopus neurons are glutamatergic. They have long, thick, and tentacle-shaped dendrites that emanate from one side of the cell body and long axons. All these neurons send their axons to various ipsi and contralateral nuclei, which are depicted in this slide. Note that the mammalian auditory system is characterized by the presence of the so-called calyces of Held and end bulbs of Held. The calyx of Held is a very large synapse named by Hans Held because of its resemblance to the calyx of a flower. This synapse has been described as the largest in the brain. Calyces of Held are formed between axons of bushy cells and neurons of the medial nucleus of the trapezoid body. Every calyx has several hundred active zones, and each active zone has about 100 glutamate-containing vesicles with several docked vesicles at a time. The calyces are characterized by the presence of many microtubules, providing stability of the synapse, regulating the distribution of the synaptic vesicles, and co-localization of the mitochondria. The end bulbs of Held are also large but smaller than calyces of Held. Astrocytes and polydendrocytes are associated with calyces and end bulbs of Held. Large synapses guarantee quick and secure synaptic transmission and provide tight coupling between acoustic events and brain activity. Dorsal cochlear nucleus has a laminar organization and belongs to the so-called cerebellar-like structures. In non-human mammals, it possesses 8 types of neurons that form 3 layers. But studies of dorsal cochlear nucleus in humans suggest the presence of only 2 layers and fewer neuron types, namely an outer, narrow, lightly stained layer and a deeper, broader layer with many cell stomata. The outer layer, consisting of nerve fibers and sporadic neurons, corresponds to the molecular layer of other species. The deep layer is likely to correspond to the intermediate fusiform cell layer and the deep polymorphic layer of non-humans. The fusiform or pyramidal and giant neurons, glutamathergic, are the main projection neurons of the dorsal cochlear nucleus. Their axons travel to the contralateral inferior colliculus and medial geniculate body. The fusiform neurons have basal and apical dendrites, the latter being branched and spiny and contacting the nerve fibers of the outer layer. Giant neurons have large polygonal cell bodies and several branched dendrites. Auditory nerve fibers and axons of contralateral distellate cells from ventral cochlear nucleus, synapses on the fusiform and giant neurons. Granule neurons, glutamathergic, are small cells with 2 or 3 short dendrites that give rise to a few branches with terminal expansions. These neurons integrate multimodal inputs, receiving mossy nerve fibers from many sources, including the somatosensory system, pontine nuclei, and auditory system. Their axons ascend to the outer layer where they form parallel fibers, similar to those in the cerebellum. Unipolar brush neurons are interneurons that ensure feed-forward excitation from the mossy fibers to the granule cells. Cartwheel neurons, glycinergic, get inputs from parallel fibers and send axons to fusiform and giant neurons. It is interesting that the mossy fiber and enlargement or rosettes, the granule cell or unipolar brush cell dendrites, and glial cells form structures that resemble the glomeruli of cerebellum. Moreover, in the cerebellum, the parallel fibers synapse on the inhibitory Purkinje cells. In dorsal cochlear nucleus, the parallel fibers synapse on the inhibitory cartwheel neurons that are considered analogous to the Purkinje cells. In the cerebellum, the Purkinje cells synapse on the projection neurons of deep cerebellar nuclei. In dorsal cochlear nerve, the cartwheel neurons also synapse on the projection neurons. The dorsal cochlear nucleus not only transfers audial information to higher CNS centers, but also plays an important role in coordination with other systems, vestibular, visual, somatosensory. To be continued.